A1.2 Nucleic Acids. How does the structure of nucleic acids allow hereditary information to be stored? And how does the structure of DNA facilitate accurate replication? So our first question is, why do offspring tend to look like their parents? Well, DNA is the genetic material of all living organisms. It stands for deoxyribonucleic acid, and DNA is the hereditary material in all living organisms. It contains genetic instructions for development, functioning, growth, and reproduction of all known organisms and many viruses. Note, this is a visualization of SARS-CoV-2, which is the RNA virus that causes COVID-19. Note, it looks a little bit different than DNA, if you've seen DNA before. DNA is a double helix. RNA is just single-stranded. So you can see here, it's just a single strand of genetic material. Now, it doesn't break this statement that DNA is the genetic material of all living organisms because viruses are not considered to be living. Because they cannot reproduce without a host, we do not consider viruses to be alive. Now, DNA and RNA are both made up of building blocks called nucleotides that are made up of three parts: a pentose sugar, a phosphate, which is negatively charged, and a base that contains nitrogen, one of four bases. Now, the bonds within the nucleotide are covalent bonds. This is a picture of a generalized nucleotide. You should be able to draw this. Make sure that you draw a pentagon with a point facing up, sort of like a little house, and the walls of the house pointing in towards the bottom. The base needs to be bonded to this exact point on the pentagon, as does this bond that leads to the phosphate. This one should go up at this angle, while this one is horizontal across to the side. Now, these building blocks, nucleotides, are joined together using sugar-phosphate bonding. Now, this makes sense because it's the bond between the sugar of one nucleotide and the phosphate of the next. So this is a sugar-phosphate bond, and it is also a covalent bond, just like these bonds within the nucleotide. And so, because we've got all these very strong covalent bonds, this forms a sugar-phosphate backbone. Sugar, phosphate, sugar, phosphate. And it's going to provide a very strong structure for DNA. Now, those bases, I mentioned there are four of them. You need to know the full names, and you need to know their base pairing rules. These bases are complementary to each other: thymine and adenine. Cytosine and guanine are also complementary base pairs. Now, they're complementary because they will only bond with each other because they only want to form a certain number of hydrogen bonds. Now, because C and G want to form 3, A and T want to form 2, they're only going to bond with each other. They're just not going to be molecularly attracted to any other base other than their complement. A good way to remember this is to remember that A bonds with T. I just think of apples in a tree. C goes with G, cars in the garage. Note, you don't have to memorize the number of hydrogen bonds, that there's 2 versus 3. Just know that there's a different number, and that's why we have certain base pairing rules. Now, RNA shares 3 of the same 4 bases as DNA. The difference is, where in DNA adenine binds with thymine, in RNA adenine binds with uracil. DNA is a double helix, RNA is single-stranded. DNA has cytosine, adenine, and guanine in addition to thymine, while RNA has cytosine, adenine, and guanine in addition to uracil. Information is stored and transmitted through DNA and RNA by the sequence of the bases, which order they are in. Right? The prefix mono means one, and the prefix poly means many. So a monomer is one molecule that can be bonded to other identical molecules to form a polymer. A nucleotide is a monomer, sugar, phosphate, base, and DNA or RNA is a chain of nucleotides that we then call a polymer. Nucleotides get linked together. That sugar-phosphate bond is formed through a condensation reaction. 2 molecules are condensed together to make this one single larger molecule. Now, one thing to remember is that a water is produced through this reaction. And just think of condensation when you have a glass of ice water on the counter. Condensation is going to happen as water vapor in the air condenses into liquid water on the outside of your glass. So just think of condensation producing water. Okay, this is how you would draw a diagram of a polymer made up of 4 RNA nucleotides. This is a drawing skill you should be able to produce. Note that each bond needs to be in this specific location. So I recommend when you draw a nucleotide, always start with the pentagon because everything is going to be based off that pentagon so that all of your angles are at the correct location. One third and final difference that we're going to talk about between RNA and DNA is the sugar. Both DNA and RNA have a pentose sugar. It's 5 sides. Os referring to a sugar, pent referring to 5. But RNA, which stands for ribonucleic acid, has ribose sugar. It's right there in the name. These are both nucleic acids, but this one is ribonucleic acid with a ribose sugar, while DNA is deoxyribonucleic acid. And so following the same pattern, we can identify that DNA is going to have a sugar called deoxyribose that has 1 less oxygen compared to the ribose sugar. So here's what each of those look like. Ribose here you can see on carbon 2 has an OH, a hydroxyl group, while this carbon 2 just has an H. Everything else is identical. So deoxyribose is a sugar found in DNA and it has 1 less oxygen than ribose found in RNA. DNA is a double helix. That's the name of its 3-dimensional shape. A helix is that spiral staircase shape. And so it's a double helix. There's actually 2 spirals. They're made of 2 antiparallel strands of nucleotides with 2 strands linked by hydrogen bonding between complementary base pairs. There's a lot of good info in there that you'll need to know. So here's DNA. It's double-stranded. We just focused on a single strand, which can either be half a DNA or RNA. Now they run anti-parallel to each other, meaning they run in opposite directions. So in this case, you can see all of these pentagons are pointing up, all of the phosphates that are associated with the nucleotide are pointing up, and the opposite is true on this anti-parallel strand. It's parallel, but it's going the other direction, anti-parallel. Notice all of the pentagons facing down, the phosphates are facing down as they're associated with a particular nucleotide. This is an important drawing skill. You should be able to draw DNA as a double helix made up of 2 antiparallel strands of nucleotides with 2 strands linked by hydrogen bonding between complementary base pairs. So when drawing this, I recommend drawing 1/2 of it here, then turning your page upside down and drawing the other half so that you make sure that you get that antiparallel going the opposite direction. You don't want to have like an extra phosphate here because we know the phosphate associated with the nucleotide is there, and so we don't want to have extra ones. For this drawing skill, if you're asked to draw it on a test, 2 rungs of the DNA ladder should be fine. You need to make sure that you write out the base pairs, the full word, cytosine and guanine. They can be on either side as long as those 2 are together. Adenine and thymine the same. Now, the bonds here are hydrogen bonds. Again, you don't have to know the number of hydrogen bonds, and so I usually just recommend my students to have a 1 dashed line so that you don't accidentally get them mixed up and then lose the mark. But if you just have 1 dashed line throughout, then that is completely fine. It recognizes that you know that's a hydrogen bond and it should be labeled as such. Now, hydrogen bonds are much weaker, and so we're going to see that DNA is going to unzip between these 2 bases. Remember, all of these other solid lines are strong covalent bonds. These are going to be our weaker hydrogen bonds. Now, note for this drawing skill, this is generalized with a pentose sugar when we talk about it in general for DNA or RNA, but since we're drawing specifically DNA, we need to specifically say the name of the sugar, which is deoxyribose. So you'll want to label the phosphate, the deoxyribose. Sometimes I like to label the sugar phosphate bond or a covalent bond, but absolutely make sure that you have a dashed line that you have the hydrogen bonds labeled. Now note, if you were asked to distinguish between DNA and RNA, you can give your answer on an IB exam in a table as long as you have this row or column, I suppose, right here, where you indicate. You can't just say DNA 2, RNA 1 without indicating what those numbers represent. So number of strands, and you need to make sure that you're comparing the same thing in each row. Alternatively, you can write this in paragraph form, but you need to make sure that you're doing direct comparisons, which means that you have to state both DNA and RNA in each sentence, and you need to say, what is it about DNA versus RNA where you're comparing the same factor? So for example, if I didn't want to do a table and I wanted to write this in sentence form, which both are perfectly correct, you could say DNA has 2 strands while RNA has 1 strand, period. Next sentence. DNA has deoxyribose as its pentose sugar. RNA has ribose as its pentose sugar, period. Next, DNA has cytosine, guanine, adenine, and thymine for its bases, while RNA has cytosine, guanine, adenosine, and uracil, period. You would get all 3 marks. Note that we said both of these words in each sentence and made direct comparisons for each of those rows of information. We've already covered this one, complementary base pairing, based on the number of hydrogen bonds that form between bases. So because we have those weak hydrogen bonds running up the middle of DNA, that's going to help us understand how DNA can replicate. When DNA replicates, those hydrogen bonds are going to be broken as the DNA double strand unzips. Just like a zipper, all these weak bonds break. And then free nucleotides that are just floating around, they're coming from digestion of other foods that you eat, are going to match up with their complement. And when that happens, we're left with 2 strands. They're identical to each other, and we're also identical to the parent strand. These are both exact copies of the strand we started with, and we call it semi-conservative replication because each new strand consists of 1 parent strand and 1 new strand. Now you might think, what? We only have 4 different nucleotides, and yet we have this incredible amount of genetic diversity that makes frogs different from bacteria, different from fungi, different from humans, and it's all just based on 4 building blocks. But if you start to think about how many possibilities there are with even just a short number of bases, it's pretty mind-boggling, and it really shows how you can have limitless possibilities of creating genetic information using just 4 building blocks. Let's look at it mathematically. There are 4 possibilities for each base. So if there's just 2 bases, we're going to have 4 squared. So we have 4 possibilities for each of 2 places. So there's 16 possible combinations. If there were 3 bases, there's 4 to the 3rd power, 64 possibilities. So you can see here the pattern is with n bases, there are 4 to the n possible sequences. With just 10 bases, we are already over 1 million possibilities. And the average gene length is around 29,000 base pairs, but it can be any length, and therefore the capacity for storing information is truly limitless. How do we get all of our DNA to fit into a microscopic nucleus? This tiny volume, if you were to unstretch the DNA in a single human cell, it would be about 2 meters or 6 feet long. But DNA is super thin. The diameter is just 2 nanometers. And that's really going to help allow for the DNA to be crunched up super tightly into this tiny nucleus and be able to store information economically. Conservation of the genetic code across all life forms as evidence of universal common ancestry. All living organisms use the same universal genetic code, all viruses with a few minor exceptions that use RNA instead of DNA, and this DNA throughout all living things is evidence of universal common ancestry. We are all related to each other, and we all have a common ancestor that used DNA. And so over evolutionary time, that DNA stuck around as the way that we pass on hereditary material as organisms began to go through natural selection and evolve into the variety of life we observe today. A quick approach to learning, thinking skills, evaluating the role of emotions and attitudes in science. I really like this one. This is Marshall Nuremberg. He was awarded the Nobel Prize in Physiology or Medicine in 1968 for his work on the genetic code. And I urge you to just pause the video for a second and read through his quote. This just shows a very emotional response to the discovery that all living things share the same universal genetic code. And it really, I think, breaks a few stereotypes of scientists being really cold and just, by the book. And it just gives a very human side to just how we felt when he made this discovery. And I find this quite profound, and I hope that you take the time to read it and just think about the fact that we're all humans and we all have emotions and we all connect with the planet in our own ways. This brings us to the end of SL content for A1.2. So if you're an SL student, you could stop the video here. If you're an HL student, we have some more to go. If we want to be able to talk about the directionality of RNA and DNA, we need to assign each end of the molecule a name. And the way we're going to do that is based on the carbons in the sugar. Now, this pentose sugar has 5 carbons. Up here at the top, again, if this was a little house, it'd be right here at the tip top of the roof, is actually an oxygen. And then this here is a carbon. Now you don't see a C like we saw on an earlier slide. This is just a shortened form because all living things are carbon-based. When we draw molecules in living things, in order to just have it be a little bit less complicated looking, we use this shortcut of having an angle represent a carbon. We just do this for carbons. We also then have assumed hydrogens in order to make every carbon have 4 bonds. But for now, just know that each of these angles here represents a carbon. So we're gonna call this carbon 1. This is carbon 2, 3, 4, and then there's a carbon 5 right here that's then bonded to this phosphate group. So we're just gonna count our carbons clockwise from this oxygen. So this is an oxygen here, and there's carbon 1, carbon 2, carbon 3, carbon 4, and then carbon 5 is here attached to this phosphate. So looking at this DNA model, we're going to see here that the end that has a carbon 5, we're going to call that the 5' end. And this side that has the carbon 3, we're going to call the 3' end. And this is how we can discuss the directionality of RNA or DNA. Now note, this is antiparallel. And so the side that has the phosphate, where the 5th carbon is the 5' end. And so when we look at the opposite side, this antiparallel side, we're going to see it's the other way around. Here's carbon 5 attached to the phosphate, so this is the 5' end, and this is the 3' end. Now note that this gets a little bit tricky when we do DNA replication. The first step is going to be unzipping that DNA strand, but DNA replication is always going to occur in a 5' to 3' direction. Now that's going to be really easy on one of the strands, because as the unzipping occurs of the parent molecule here in dark brown, we're going to be able to add nucleotides one by one by one in a 5' to 3' direction on the leading strand. No problem. But we're going to see that we're going to run into some problems, because on the lagging strand, we can also only go 5' to 3', but that moves us away from this replication fork. And so what we're going to see is that the DNA gets copied in fragments called Okazaki fragments. And so we'll have 5' to 3', and then another fragment, 5' to 3', and so on as the replication fork opens to the right. We are going to classify our bases as either pyrimidines or purines. Note that pyrimidines have 1 ring of atoms in their base, while purines have 2 rings of atoms. Note that a pyrimidine and a purine will always be bonded to each other. That is going to allow DNA to have a stable width. If we had, for example, 2 purines, 2 double-ringed bases, the larger size, then DNA would not have a steady width. It would go out and get wider at that point. Similarly, if we had 2 pyrimidines, it would go in and get narrower at that point. But that would decrease DNA stability. And so by having a smaller pyrimidine always bond with a larger purine, that's going to give us higher stability and a consistent width throughout the entire molecule. Now, how do we remember which one is which? This one's not my best mnemonic, but maybe it'll work for you. So what I do is I just picture like a big, beautiful field of agriculture. And I just think, oh, it's so big and it's so pure. It's all this photosynthesis happening, taking all this CO2 out of the atmosphere. And then I think agriculture starts with AG. Those are my purines. They're so pure, ag, and they're so big. They're the ones that have double rings. So hopefully that one works for you. All right, so our consequence again of always having a purine bonded with a pyrimidine is that stable, equal width all the way through the three-dimensional double helix structure. So earlier we talked about how 2 meters of DNA has to fit into a tiny microscopic nucleus of a cell. And one of the ways that that is possible is that it gets packed up really tightly around proteins. You're going to see this beads on a string model, which this gray represents DNA, which is wrapped twice around an octet, so 8 histone proteins, and then it gets sealed with a single histone protein called the H1 protein. And so this DNA is getting tightly wrapped around these 8. I like to think of it as a gift, where you have like a gift that gets wrapped with ribbon and then kind of sealed in with a bow. And then there's these little bits of linker DNA that connect this next set. And each of these little beads on a string is known as a nucleosome. And it's going to help the DNA get packed in really tightly, as well as controlling which DNA is going to get turned into proteins at any given time. Now, you should use some molecular visualization software to take a look at the 3D nucleosome model and be able to turn it around. Just do a Google search for that software, but it is something that I would like you to be able to do. Next up, the Hershey-Chase experiment in 1952. This is the year before Watson and Crick came up with the DNA's structure. Martha Chase and Alfred Hershey were trying to answer the question of, is protein or DNA the genetic material? Now, this is an experiment that you should be familiar with. It's a very elegant experiment. It might be a little bit tricky to get since you're probably brand new at biology, but try your best to follow along because it's a very classic experiment that you should be familiar with. So first up, back to this question. Is DNA or protein the genetic material? From the late 1800s, it was known that chromosomes were composed of protein and DNA, but it wasn't clear which of these was the genetic material. Like we just learned, DNA is wrapped around proteins. And so since DNA and proteins are both found together in chromosomes, it wasn't clear which one is actually carrying the genetic material. Now, proteins are made up of 20 different amino acid subunits or building blocks, whereas DNA contains just 4 types, those A's, T's, C's, and G's we just talked about. And many specific functions of proteins had already been identified by this time. So for these 2 reasons, most biologists viewed protein as the more likely candidate. 20 building blocks is a lot more to work with than only 4. Variety and specificity of function were considered essential for hereditary material. And so Hershey and Chase chose a virus. This is called the T2 bacteriophage virus. It's a virus that infects bacteria. Now remember, viruses aren't considered alive. This, in fact, is a super simple structure. It's just DNA surrounded by a protein coat. And the way that this bacteriophage virus works is it injects its genetic material into a bacteria, and it then uses all of the parts in the bacteria to replicate its DNA. Now, DNA contains phosphorus, but not sulfur. And proteins contain sulfur but not phosphorus. Remember that phosphorus in the phosphate group of a nucleotide we talked about at the beginning of this video. Brilliantly, Hershey and Chase cultured some viruses that contained proteins with radioactive sulfur, thus marking those proteins. They also cultured some viruses that contained DNA with radioactive phosphorus, thus marking the DNA. They then infected separate groups of bacteria with each of those 2 viruses, one with marked DNA, one with marked protein. After the bacteria were infected, they then used a blender to separate the viruses from the bacteria they had infected. So all of those, going back to this picture all these viruses are on the outside of the bacteria. They've put their genetic information in to make copies of and then burst out and spread. So then they used a blender to knock all of these viruses off. They then used a centrifuge, and the centrifuge spins around the sample really, really quickly and separates it out by density. And all of those bacteria are more dense, and so they ended up in the pellet while the viruses were suspended in the liquid, the supernatant. And so the viruses were then separated from the bacteria, and Hershey and Chase could test whether there was radioactivity then in the pellet or in the supernatant. Hershey and Chase wanted to determine whether the virus had injected DNA or proteins into the bacterial cell. So let's look at their results. The first experiment, they tested proteins. Here's where they labeled those protein coats with this radioactive sulfur. When they allowed the viruses to infect the bacteria, we know that it's actually DNA that's being injected. And so no radioactivity entered the cells. Once they blended all those viruses off, centrifuged them to separate them, they were able to measure radioactivity in the supernatant, where all those viruses were, because the proteins had not been injected into the bacteria. The conclusion was that proteins are not the genetic material. But in the other experiment, where they radioactively labeled the DNA we know is the genetic material, and it was what was injected into the bacteria. So then they blended it all up again, centrifuged, separated everything out, and they measured the radioactivity now in the pellet, where all of those more dense bacteria were, allowing the conclusion that DNA is the genetic material, not protein. Quick nature of science moment. Technological developments can open up new possibilities for experiments. When radioisotopes were made available to scientists as research tools, the Hershey-Chase experiment became possible. Lastly, Chargaff's data on the relative amounts of pyrimidine and purine bases across diverse life forms. Now, if you pause the video and take a quick look, see if you can find a pattern for the base composition for each of these 5 organisms. Note that for each of these 5 organisms, there's approximately the same percent of A's and T's as there are of G's and C's. You can see here 28, 28, 21, 21. Now, before Chargaff took his data, scientists hypothesized that DNA would have a repeating sequence of 4 bases known as the tetranucleotide hypothesis. ATGC, ATGC, ATGC. This would mean that the 4 nucleotides occurred in equal numbers. And so when Chargaff analyzed DNA samples from a range of species, he was then able to falsify that hypothesis with his results. And he noticed, no, there is not the same number of each base. In fact, it varies by species, but there's this pattern where A's and T's are going to be approximately the same, G's and C's are approximately the same. And this was going to help then Watson and Crick in 1953 be able to come up with the complementary base pairing rules and eventually then the full structure of DNA as a double helix. So lastly, falsification, the problem of induction. Now, the scientific method, scientists make observations, detect patterns, and then form generalizations. We then use these generalizations to draw conclusions about things that have not yet been observed. This is known as inductive reasoning. The problem with induction is there's no certainty that the observed things will conform to the generalization. We might have a pattern, and so we can use that pattern to predict a data point we don't know, but we cannot say with 100% certainty that that is actually going to be the case. The question is then, is there anything about the natural world of which we can be certain when we have not observed all cases? So while this is a problem, we do have a certainty of what's not true, falsification. Falsifiability is the idea that in science we can at least be certain of what is not the case by finding a counterexample. And that's exactly what Chargaff's analysis did. It falsified the tetranucleotide hypothesis that the 4 DNA bases occur in equal amounts. Similarly, the Hershey-Chase experiment falsified the hypothesis that protein is the genetic material. The work of these scientists proves certainty of what was not the case, and that's what we can be sure of. All right, folks, good luck out there.